Hi, this is Roger Green, executive producer and host of the Surfing the Mesh Tsunami podcast. This week, over the holiday week, we will be sharing 5 conversations from episode 27, our coverage of last month's Deliver Meeting 2024. This conversation takes a look at one paper, one issue from papers in general, and one other session that was pivotally important. The paper comes from Jörn Schattenberg, who highlights a late breaker presented by Mazen Nuruddin on Efimosfermin alfa, an FGF-21 dosed Q4 weeks, or 12 times a year. In this trial, which was a phase 2b small population, even 6 months of therapy demonstrated significant effect both on NAS score and on fibrosis. This led to several comments from participants about why they thought this is so important in terms of compliance. To Jörn, this and other studies demonstrate the place FGF1s are likely to have in MISH therapy. The other studies coming from Efruxifermin and Pegozafermin. This led Mike Betel to ask the question, which is, why do placebo groups score so high successfully in clinical trials? The answer seems to be that the diet and exercise suggested for patients in general is so well enforced in clinical trials that patients are more compliant with those elements, and as a result, they do better. This led to Mike wondering what would happen if we could get that kind of behavior every day in life, an issue I'm sure we'll be talking about for years to come. The second session covered in this conversation was from Michelle Long, who praised the FDA Corner session. Particularly, she noted the agency was transparent on what it's looking for and willing to engage industry and academia as partners in finding paths to solve specific problems, most important of which is moving away from biopsy. She expresses optimism that the scientific community will come together to address these issues in the months and years to come. As you listen, you'll hear all of our enthusiasm, which was infectious within ourselves, and we hope to those of you who are listening to this episode.
Jörn Schattenberg.
The one paper I wanted to highlight was also in the late breaker presented by Mazen Nuruddin, and it's Efimosfermin alfa drug, which is a phase 2 trial. And the interesting aspect here is that this is from the FGF-21s, which are engineered and pharmacology altered to have an extended half-life. And this drug was actually just injected every 4 weeks. And I think this is something that we haven't seen before, so it's a new level. And actually, the primary endpoint in that trial was after 24 weeks. The patient received 6 doses of drugs, and that's not a whole lot of drug injections. So I felt it was very innovative, or at least very interesting from that aspect. The study, again, phase 2, 84 patients randomized, 24 weeks treatment with biopsy as a readout, 2 arms, 300 milligrams efimosfermin versus placebo, showing superiority in MASH resolution and 1 stage in fibrosis improvement. And I found that was pretty, the numbers are difficult to compare to the phase 3 trials. Numbers for MASH resolution were 68 versus 29%, and fibrosis improvement 45 versus 21%. Additional markers, MRI, PDFF, and so went in the right way, biomarkers. So just the concept that we can treat this with these FGF-21, which have shown by other companies shown robust effects, where we keep on seeing this, and prolonging the treatment to 4 weekly, I felt was very interesting here.
Roger Green.
Yeah, Jörn, I had a similar reaction to that paper. It feels I haven't seen data on this, but intuitively it feels to me that the 2 easiest compliance schedules for people to live with are once a day and once a month, even easier than once a week. And to be able to get to a Q4 week schedule with a drug that has a measurable effect as quickly as 6 months kind of solves 2 issues at once: easy for patients to tolerate and relatively quick to figure out what's going on. So I thought that was a pretty fantastic result. I agree.
Mike Betel.
Hey, Jörn, it was shared too, because I made a note of the same study. Combined, I guess that's MASH resolution and fibrosis, 39% versus 18% also. So I guess when you look at everything combined.
Yes, so the metrics all went in the right way. I think, again, those numbers they're statistically different from placebo. We heard the phase 3 trials, and the numbers normally go down if you study more patients and different durations. So, but it, ticked the box. I think there's a rationale to carry this forward. I mean, with this class of drug, the emergence of drug antibodies is sometimes a concern, and we also saw more extended data being released, 96-week data on the Akero compounds, for example, and they still had efficacy. So it felt like there was room for the FGF-21s that haven't really taken off in diabetes, but seem to be really impacting on the liver. And that's reassuring, adding yet another MOA we could combine or choose from, assuming we'll learn in the future that maybe a certain genotype is not so sensitive to certain MOAs.
I have a group question, if it's okay. Why do you think that the placebos have done so well on all the studies? So these are patients, is it just the behavioral support being involved that you have to check in with the clinical study team? Or
Yeah, I'll give you my quick impact or view on that, because I think it makes a huge difference if you're coming back to a study coordinator that puts you on a scale, asks you about dietary and nutrition, shares some values with you. It's a biofeedback you normally don't get, and as such, it's much more intense. Of course, those are simple measures we could do in clinics, but it's not happening at the same extent. And so clearly being cared for by a team that's authentic, that cares about you, that spends time with you is something that adds to that placebo response.
Jeff McIntyre.
Yeah, I'll second that in saying that it's a larger scale measurement of what a lot of us experience on a more popular scale or general scale, meaning that, if I count my steps, then I'm more likely to get my steps in. And so I think that awareness, and especially if you add the notion of support to it, clinical support, we see this across all levels of patient engagement, that whether it's, and it's part of why we talk about, clinical trials as healthcare for patients, especially when they're in areas where there's no medication, is that, you see it from the clinical side, from clinical support. You'll see it in support groups where people can be a little more engaged. You see it, if it's counting your steps or doing something like that. And there's certainly a way to go overboard with that. But I think just having that kind of regular, casual, just kind of update that lets you know is one of the reasons why placebo care has been so helpful in this. And I'll say the other reason I think is because it has emerged in an area where there's otherwise been a vacuum of support. That it just is the choice for people is don't do anything, sit on the sofa, watch streaming, ignore the symptoms, wallow in the stigma. And so then suddenly to actually have an engagement and to move forward in a place actively, I think is a tremendous benefit, and why we see some of those results.
like, how cool would it be if we could gather all of the study data with that, even on the placebo arm, and just apply some of that to the real world to teach just the day-to-day treatment that patients are getting, just some of it. It would be obviously a big win, right?
I'm down.
We're all down. Okay. Who would like to go next? Given the time is flying and we're having a great conversation, but we've only heard of 2 or 3 papers so far. So
let me volunteer Michelle if she's willing, because I think Mike and I are going to have a very similar session to talk about, actually. So.
Michelle Long.
Yeah, great. A lot of really great papers already discussed, but I would say that I'll talk about a session. Also, from a drug developer point of view, which was highly appreciated. This was the FDA Corner, where the FDA or representatives from the FDA presented on a number of topics, and then allowed time for question and answer. So one thing that was presented was they really walked through the Rezdiffra approval and what were some of the considerations there. Of course, a lot of these documents now are in the public record and one could go through and look at them, but I think the average clinician doesn't. And so I thought it was really great and just really commended them for being so transparent to talk about what are the considerations, because it is really challenging, I think, to make sense of these data, especially, we talked a little bit about the placebo response. Of course, there are real biologic differences and benefits for being in trials, but a lot of the placebo response probably also has to do with the histology reader setup. It can be quite complicated. And then, of course, these studies take a while to do, and we're learning along the way. And so what might be best practice at this moment in time is impossible to implement 3 years prior, but yet those are the data then that you interpret. And it was really nice to see how they handled the Rezdiffra data, and also how they went through and, showed how no matter how you looked at it, they showed consistency of results, which I think was very, reassuring to them that the effect was there. So that was super interesting. And then they also went on and talked about accelerated approval and made clear about, okay, you have now you have Rezdiffra that has been approved via the accelerated approval pathway, Perhaps there'll be other drugs in the future, and really made clear, though, that they need to see this substantial evidence of efficacy and demonstrating clinical benefit, and really explained in detail as to what that would look like or what that could look like if that's not achieved. So it's still a tall order, and there's a lot of work to be done to maintain these trials to clinical benefit. And then finally, in this session, we've heard now over the last year, and I've also talked about this on the podcast, that there is a real momentum around kind of a new level of transparency that the FDA is giving, at least on their personal views of how they look at these endpoints and how they look at biopsy-free potential trial designs. And so there was a lot of clarity, I think, given around what a reasonably likely surrogate endpoint looks like. This is, of course, what we consider the histology endpoints today, that they're reasonably likely to predict the clinical benefit. But of course, with all the emerging data, it's obvious that there are some other potential candidates out there. And so it was great to hear the agency come out and say, yeah, we hear you. Let's look at those data together and let's see what that, could look like. So there seems to be, a willingness to have those conversations. It was nice to hear, that they don't need to see these trials running to clinical benefit. They're willing to have these conversations now to help inform the future trials. Also, back to your original point, Roger, about how do we innovate in trial design, meeting the innovations that we have in the clinic of moving away from doing biopsies to identify the treatment population. Can we bring that into trials moving forward? So I think it was a really helpful session. I think a lot of people were happy with that degree of transparency, and so I think we've also learned it's not something we're all going to just sit around and wait to happen. We as a scientific community need to come to the agency with a proposal. And that was also made super clear. So I think it's obvious of what needs to be done. And the question is, how do we as a scientific community come together around those ideas and present something that makes sense and see what the potential is and what the implications are. So I have high hopes for the future. I think there's work to be done, but a plan that I think is forming. So it'll be really exciting. I know part of what you asked, us to think about was, what does the next liver meeting look like? I really think we'll see a lot more clarity around this over the next year.
If I can add to that real quick, I really appreciate that. And that was an amazing session. And Michelle and I have talked, offline about this quite a lot as well. I would add to this that there's a lot of momentum. There's a lot of interest in this.
And now back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please put them in the review section of the page from which you downloaded this conversation, or send an email to questions@surfingmash.com. Over the rest of the holiday, we'll be sharing conversations from episode 25, in which Maru Ranella went over expert recommendations on use of resmetirom, and also a conversation Jörn and I recorded with Scott Friedman last summer that never made it to air. Plenty to listen to while you're at home and away. And we'll be back in January covering MASH-TAG and then moving into our new 2025 Surfing Mash V2.0 format. Take care, stay safe, surf on. We'll see you on the podcast. Bye-bye now.